All right, so hopefully you guys have had time to look over, read through, maybe do the first part of the lab on mitosis. You're comfortable with it because now we're going to move into sexual reproduction. And so in humans, we have what are called somatic cells. And somatic cells are all the cells in your body except for your sex cells. So, except for your sperm and eggs. We're going to learn how we make sperm and eggs in this part of the lesson. So, in those cells, there are 23 pairs of what we call homologous chromosomes. That means one chromosome's from dad, one's from mom. They pair up, they have the same type of information on them. And we also have what are called the sex chromosomes. So, that would be chromosome pair number 23. Chromosomes 1 through 22, we call those autosomes. They're the same in males and females, basically, but then we have the sex chromosomes that will determine whether you are a male or female, genetically, a male or female. So if you have two X chromosomes, you are female. If you have an X and a Y, you are male. Now, what I had mentioned with the homologous chromosomes is that they are very similar. They have the same length, their centromeres are in the same position, they have the same genes in the same location. Now, the type of gene they have may be different. So, mom might be short and dad might be tall, and mom might have brown eyes and dad might have blue eyes, but they'll have that information in the same spot on that chromosome. It's these differences in these forms or versions of the genes that make us all different. So what we're going to do in meiosis is we're going to take what's called a diploid nucleus and turn it into a haploid nucleus. So diploid means they have both mom and dad's versions of each gene. Haploid means you either have moms or you have dads. You don't have both. And so we see this in how we actually make our eggs and our sperm. So eggs and sperm, besides being called sex cells, are also called gametes. Now, fertilization is where we have the egg and the sperm come together, and they're going to produce a fertilized egg, which is referred to as a zygote. So here we have quite a bit of vocab for you. So here's our diagram. Of course, in the ovaries is where the female will make her eggs. The testes would be where the male makes the sperm. We notice that our cells, all the other cells in our body are diploid. They have 46 chromosomes, but our eggs and sperm only have 23. So when the 23 and the 23 fuse together during fertilization, we then have back our diploid zygote. So we're back to 2N. And now that fertilized egg is going to undergo mitosis to form that human being. So meiosis and mitosis are both preceded by duplication of chromosomes. So they both go through that cell cycle. They go into interphase. They make that copy of DNA. But meiosis has two cell divisions and mitosis only has one. Because in meiosis, we have that duplication of our chromosome, it then follows those two divisions, one in which to split up the mom and dad's um, homologous chromosomes, and the second part is to split up what we call the sisters. So normally, again, I would be drawing meiosis out on the board, and I have that in your lab book, and I also have that on one of my videos for you to watch, plus all the other videos that I've put on you there for you to watch. So when we look at the diagram, I'm going to kind of go through this a little bit fast. We have our interphase. Notice our DNA is unwound. It's called chromatin. We have our centrioles again. They're going to be in there kind of managing all those chromosomes. We have prophase 1. Notice we have to use the 1 or the 2 because there's two cell divisions. So in the first cell division, we're back to our PMAT1. Prophase, metaphase, anaphase, telophase. The same types of things are going to happen in those phases that happened in mitosis. So in prophase, the DNA is going to wind all up into chromosomes. We're going to lose our nuclear envelope. And then in metaphase, we're going to line up. Now notice we are not lined up single file. We are lined up facing our homologous chromosome. So mom's chromosome 1 finds, and this is a lot. There's a lot of things going on here in prophase. Notice how I do have the chromosomes are paired up. So mom's big chromosome is going to find dad's big chromosome, and mom's medium is going to find dad's medium, and the small and the small. And they're going to get together, and then you may also have this thing called a crossing over. So notice right here, the two arms are crossed over, and they may actually split and swap sections of the chromosome. And this causes, so this is the first place that we're going to get some genetic variation. So remember, in mitosis, we ended up with 2 identical cells. In meiosis, we're going to end up with 4 genetically different cells. And so this is our first area of genetic variation, is in crossing over. The second one here is on how we line up on this metaphase plate. Now notice I have moms, 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 okay, dads, dads, dads. Now I could have moms all on one side, I could have dads all on one side, I could have this one flipped. This is completely independent. So this is called independent assortment. So it does not matter how, on what side they're on that plate, as long as they're facing their partner. Now this will also give us our second area where we get genetic variation. And then, of course, anaphase. In anaphase 1, we're not splitting the sisters, we're splitting the homologous chromosomes. So mom's going one way, dad's going the other. I refer to this as the divorce because we're splitting up our parents. And this is where our cell is going from diploid to haploid as we split those parents up. Now we have a problem in that, yes, we've split up our parents, we've gone to haploid, but we still have duplicated chromosomes. We still have those sisters. So now we have to get rid of the sisters, and this is going to look just like mitosis. So again, we have our telophase 1 and our cytokinesis. We're going to split into 2 cells. We are, if we started to reform our nuclear envelope, we're going to break that down. We are going to line up in the middle on the metaphase plate. And then we are going to split the sisters apart in anaphase 1, or sorry, anaphase 2. And then we're going to split up our cells and our cytoplasm in telophase 2 and cytokinesis 1. Sorry, cytokinesis 2. And then we will have 4 cells when we get done. So I wish they would show that in that diagram, but they don't. So let me try to go back. So here would be 1, 2, 3, 4 unique cells, 4 unique haploid cells. So this would be making, again, our eggs and sperm. So here, mitosis and meiosis, they both begin with diploid parents. They both have their chromosomes duplicated in interphase. The end, however, is different. In mitosis, we have 2 genetically identical diploid cells. And in meiosis, we have 4 genetically unique haploid gametes. So here's a side-by-side comparison here. So we can see how we started off here. This is our cell. We're saying that our diploid number is 4. We have 4 chromosomes. Notice we're going to replicate in interphase both of them. We make the sisters, but in this first phase here in meiosis, not only did we make the sisters, but the pairs partner up, and we might have synapse is another term for crossing over. Right, so they pair up, they line up in the middle, versus mitosis, they line up single file down the middle. We're going to split our sisters apart. We have 2 identical cells. Notice they look just like the original parent. In meiosis, however, now we got to go through our second cell division to split the sisters. And now we have 4 cells that are haploid. So notice we started with 4, and then we made our copies. But we still, what we're going to do is we still have 4 when we get done. We started with 4, we end with 4. Here we started with 4, we ended with 2, because we split the mom and dad apart. So again, diploid, haploid, sexual reproduction, asexual reproduction. I already mentioned independent assortment, so this is kind of showing you a side-by-side comparison. I could have put dads all on one side, I could have put moms all on one side, or I can flip them. I can flip them the other way. So there are multiple combinations when you have, think about it, 23 pairs down the middle of how they can be on different sides to get you different variations of those eggs and sperm. All right, so homologous chromosomes again have different versions of genes. One version is inherited by your mom, one is from your dad. We'll talk about which one's dominant, recessive, and how that influences what you look like in our genetics unit. But right now, just know that mom's copy goes to one side of the cell, dad goes to the other during that anaphase 1. So here we show a dominant mouse. It has both black eyes and dark fur, versus this mouse has the red albino phenotype, which is what they look like. And so we can see here we have, if we split, the red chromosome goes to one side, all of those eggs and sperm will get the big uppercase or dominant genes, versus this side, they're all going to get the recessive. But with genetic recombination, we can get some crossing over. Remember where we kind of split those sisters, swapped arms, and so that's called a crossing over. So they're going to join at what's called the chiasma, which is where they basically cross. We also refer to that again as a synapse. And we're going to exchange those parts. So we can see here, here's where they line up, when they're paired up during that prophase 1. And here we're going to have a crossing over event. There's the chiasma or the synapse. And now when we separate out, notice how we have not just dominant, but now we have a recessive dominant, recessive. So these are the 4 different possible gametes, so the different combinations we can get in our eggs and sperm, which would lead us to different traits or characteristics being showing up in their children, in their offspring. All right, the last part of this presentation has to do with what happens when things go wrong. So I would prefer if you guys at this point stopped, read through, got comfortable with meiosis before I start talking about problems, maybe do the lab, and then come back to the next part of this lecture.